So I will start with the commemoration of our past member, Har Gobind Khorana. I just follow my notes. He died pretty precisely 1 year ago, in the 9th of November 2011, at an age of 89 years and almost 90 years old. He had been growing up in British India at that moment in the 1920s, went to the university in Lahore and obtained a Master of Science with specific interests in organic chemistry and biosciences. He then left his home country, going to Liverpool in England for a PhD study, and in the late 1940s he went for a first postdoc to Switzerland at the ETH in Zurich, where he stayed, I think, roughly 1 year. If I'm right, I'm not quite sure, but I think that that was the moment that he encountered his future wife, and they got married, because I have a memory that she was of Swiss grown up. It's correct? Okay, good. Yes, so I have his link with that. He from the ETH in Zurich, he went back to Great Britain, to Cambridge, where he deepened his interest in organic chemistry, analysis and synthesis, and so on. And he went later on, 1952, to Vancouver, where he stayed for a number of years before getting a professorship at the University of Wisconsin in Madison, where he deepened his investigations on the genetic code. 1960, at that moment, of course, the question was still unclear how the sequence of nucleotides are read and finally translated into gene products, which usually are proteins, occasionally also already at the level of RNA molecules. Khorana was a specialist in nucleic acids rather than in proteins, and of course the detection of the genetic code on which to which he made very important contributions is a matter on how from the nucleic acids, first of all, RNA is copied, and how then the nucleotide sequence on the RNA are translated into a sequence of amino acids in the nascent protein. As we all know now, there's a triplet code, so that each sequence of 3 nucleotides has some particular meaning. And he made important contributions to that, and other scientists, in particular Marshall Nierenberg, who is also a late member of our academy. He died just a few years ago, who also contributed essentially to that, and also Robert Hawley. These 3 scientists received for their discovery the Nobel Prize in Medicine in the year 1968. So you see the investigations were done in the early 1960s, the essential ones to the middle of 1960, and very rapidly they obtained the Nobel Prize because it was obvious at that moment to everyone that this was a very important, fundamental insight into the laws of nature. I may add here that, interestingly also, it turned out that this genetic code is a universal code. All bacteria, all plants, all animals, including us human beings, use the same genetic code. There are major alterations which I personally interpret as essential from the point of view of genetic stability. This, I add that out of the contributions of Khorana, I add that I think that mitochondria, for example, and some other organelles which are in our cells and plant cells and all over, as possibilities to use energy, they have also genetic information, and the hypothesis, which is a good hypothesis, is that they stem from microorganisms, but having lost in the meantime a lot of genes which are inessential for their functions in the cells. And at that moment, it is probably appropriate that they have a slightly other language so that there is not too much exchange between these small organelles in the cell and the nucleus. So that is guaranteeing a certain independence of the genetic information in these cells. I come back to Khorana. He continued in the subsequent years his interest in organic chemistry and in applying that knowledge to biological problems. He got a new position 1970 at the Massachusetts Institute of Technology, and in 1978 he became member of the Pontifical Academy of Sciences. I had become a member in 1981, and in the following years I had nice occasions to meet Khorana and talk with him on our common interests in biological fields. In the meantime, he continued his scientific contributions to a better understanding of life. He worked on functions of ATP, of coenzyme A, particularly for the acquisition and provision of energy to the cells, and that involves also polynucleotide synthesis. He then, that led him to consider and actually trying to improve methodology for what is called total synthesis of genes. That means from nucleotides putting things together and making entire sequences. Of course, that was done by knowing the sequence which were found in nature and trying to do it by good organic chemistry, by synthesis, putting these things together in the same order as nature does. So in that way, I consider him as an important pioneer of what is nowadays called synthetic biology. He worked on, in particularly on bacteriorhodopsin, which is a protein in biological membranes and has its function to transduce light. So that is an important contribution to better understand how our eyes function. I think that's about, he finally retired from this professorship, and I just want to finish in thanking him post-mortem for his activities in our academy. I think we appreciated his presence each time when he was here and was still active in our activities.